Okay, let's dive in. Think for a second about the most dedicated worker you know. Day in, day out, year after year, never taking a break. That's your heart. Pumping blood relentlessly, keeping you alive. But how? How does this incredible, tireless organ actually do that, beat after beat, decade after decade?
It's an amazing piece of biological engineering, really. It pushes the boundaries of what seems possible. It's way more sophisticated than just, a simple pump. It's this highly coordinated system of muscle electricity mechanics.
Exactly. And we've got a stack of sources in front of us today that kind of peel back the layers, really getting into the intricate physiology. This isn't going to be a surface skim. We're aiming to understand the heart's function with the depth, the insight that the pros rely on.
Yeah, our focus today is a deep dive into the physiology of cardiac muscle, understanding precisely how the heart operates as a pump and the absolutely critical function of its valves, all based on the material we've got here.
Right. We're here to unpack the complex mechanisms, maybe find those genuinely surprising aha moments, and highlight the key facts that make it all possible, giving you that solid foundational understanding. So let's start with the fundamental architecture laid out in our sources.
Okay, well, the material makes it clear. Don't think of the heart as just one single unit. It's really two essentially separate pumps operating side by side, and they're enclosed within that protective sac, the pericardium.
You've got the right heart, and its job is pushing blood through the lungs, right, for oxygenation.
Exactly. And then there's the left heart. That's the high-pressure pump, propelling that oxygenated blood out to the entire rest of the body, the systemic circulation.
And on each of these sides, you've got two chambers, an upper one, the atrium, and then the lower, larger ventricle.
And the atrium on each side, it acts mostly as a primer pump. Contraction isn't super strong, but it plays this important role in helping to push that last bit of blood into the ventricle on its side, kind of topping it off.
But the real heavy lifting, the force for pumping blood either to the lungs or the body, that comes from the ventricles. They're the powerful workhorses.
Absolutely. And this whole structure, like I said, is held securely within the pericardium, that double-layered sac. It lets it move freely, but keeps it, anchored in place.
So that's the basic layout. What about the actual stuff it's made of, the muscle itself? It's definitely not like, say, your bicep muscle, is it?
Oh, not at all. The sources really highlight the three main types of cardiac muscle tissue. You've got the main contractile muscle of the atria and the ventricles. That's the tissue doing the actual pumping work.
Okay. And then there are these specialized excitatory and conductive fibers. What's different about them?
These are fascinatingly different. They have very few contractile units, the little myosin and actin bits, so their physical contraction is actually pretty feeble. But their superpower is electrical.
Ah.
They're specialized for automatic rhythmic electrical discharge, basically, generating these action potentials spontaneously, and for conducting those electrical signals really rapidly throughout the heart.
So they're like the heart's built-in electrical grid and its pacemaker system, setting the rhythm and making sure the signal travels efficiently.
That's a great way to put it, exactly. Now, looking closer at the atrial and ventricular muscle anatomy, the sources show it's arranged in this complex branching lattice work pattern, but despite that different arrangement, it is striated, just like your skeletal muscle.
And it uses the same basic machinery for contraction too, right? Actin and myosin filaments sliding past each other.
Yeah, that core mechanism is shared, but here's a fundamental difference that's really key to how the heart functions as this unified pump. It's the concept of the syncytium.
Okay, tell me about the syncytium. What exactly is it and why is it so important for the heart?
Right, so when you look really closely at cardiac muscle cells, you see these dense lines separating them. They're called intercalated discs. These are basically the cell membranes. But, and this is the crucial part, at these discs, the membranes actually fuse together to form these permeable connections called gap junctions.
Like little tunnels or pipelines directly texting the insides of the cells.
Precisely. Ions, charged particles can flow really rapidly and easily through these gap junctions. So because of this, when an action potential, that electrical signal, starts in just one cardiac muscle cell, the signal, carried by those ions, spreads almost instantaneously through the gap junctions to all the cells connected to it.
Wow, so the entire mass of connected cells acts like a single functional unit. That's the syncytium.
Yes, it truly functions as one. And the heart actually has two such syncytia. There's the atrial syncytium, which makes up the walls of both atria, and then there's the ventricular syncytium, making up the walls of both ventricles.
But these two big syncytia, the atrial and ventricular ones, they aren't directly connected electrically, are they?
No, and this is absolutely critical for the heart's timing, something professionals grasp immediately. The atria and ventricles are separated by a layer of fibrous tissue, kind of like an insulator, right around the openings where the AV valves sit. Electrical impulses normally cannot conduct directly across this fibrous tissue.
So the electrical signal has to take a specific detour to get from the atria down to the ventricles.
That's exactly right. It travels only through a specialized conductive pathway called the AV bundle. This deliberate structural separation and the required detour through the AV bundle is what ensures that the atria contract just a little bit before the ventricles do.
And that timing, that slight delay, is essential for optimal pumping efficiency. It lets the atria prime the pump, essentially, loading the ventricles before they squeeze.
Absolutely vital. Now, speaking of those electrical signals, the action potentials in cardiac muscle are really quite distinct, especially when you compare them to skeletal muscle.
Yeah, the sources point out that really long plateau phase you see in ventricular muscle. It averages about 0.2 seconds, right? Skeletal muscle action potentials are way shorter.
Much shorter, yeah. Skeletal muscle action potentials are brief spikes. They're mainly driven by fast sodium channels that pop open and then slam shut really quickly. The membrane potential shoots up positive and then rapidly comes back down, repolarizes.
But in cardiac muscle, after that initial spike, the voltage kind of hangs out up there, stays positive for a significant amount of time.
Exactly. The intracellular potential goes from its resting level, maybe around negative 85 millivolts, all the way up to about plus 20 millivolts, and then it just stays there in that plateau phase before it finally repolarizes. And this plateau is the key reason why ventricular muscle contraction lasts so much longer. The sources say up to 15 times longer than typical skeletal muscle twitch.
Okay, that's a huge difference. So what specifically causes this long action potential and that characteristic plateau?
Well, the sources highlight two main differences down at the molecular level, dealing with ion channels.
Yeah.
First, the types of channels involved are different. Skeletal muscle mostly relies on those fast sodium channels. Cardiac muscle uses fast sodium channels, plus these things called slow L-type calcium channels.
Calcium channels.
Okay, yeah. They're also sometimes called calcium-sodium channels. They open up much more slowly than the fast sodium channels, but the really key thing is they stay open for several tenths of a second. This prolonged opening allows a significant influx of calcium ions and also some sodium ions into the cell.
And this influx of positive charge, the calcium and sodium coming in, helps keep the inside of the cell positive, maintaining that depolarized state.
Exactly. That flow through the slow calcium-sodium channels is the primary driver holding up that plateau phase. But there's a second difference too, and it has to do with potassium permeability.
How does potassium fit in? I know it usually flows out to repolarize things.
It does. But in cardiac muscle, immediately after the action potential starts, the membrane's permeability to potassium ions actually decreases significantly by about 5-fold. This is quite different from skeletal muscle.
So during the plateau phase, less positive potassium is leaving the cell than you might expect.
Correct. And this reduced outward flow of positive potassium ions further helps to keep the membrane potential positive during that phase. It counteracts the tendency to repolarize too quickly.
Okay, so you've got positive calcium and sodium coming in through slow channels and less positive potassium going out. That maintains the plateau. And it finally ends when those slow calcium channels close up.
Pretty much. After about 0.2 to 0.3 seconds, those slow calcium-sodium channels finally inactivate, they close. The influx of positive charge stops. And right around then, the permeability to potassium rapidly increases again.
Letting potassium rush out and quickly bring the membrane potential back down to its negative resting state?
That's the rapid repolarization phase. The sources illustrate this conventionally in numbered phases, which is helpful for understanding. Phase 0 is that initial rapid depolarization thanks to the fast sodium channels. Phase 1 is a brief initial dip in potential, a bit of repolarization.
Then phase 2 is the critical plateau we've been talking about, sustained by that slow calcium-sodium influx and the decreased potassium efflux.
Right. Phase 3 is the rapid repolarization, when the calcium channels close and the potassium channels open wide, letting potassium out. And phase 4 is just the stable resting membrane potential, back around 80 to 90 millivolts. Understanding these phases and the ions moving during each one is absolutely fundamental to grasping cardiac electrophysiology like a pro.
Yeah, that detail is crucial. Now, speaking of electrical signals traveling, how fast do they actually move through the heart muscle itself, the main atrial and ventricular muscle?
Well, in the bulk of the atrial and ventricular muscle fibers, the conduction velocity is actually relatively slow. The sources say about 0.3 to 0.5 meters per second. That's considerably slower than large nerve fibers or even skeletal muscle fibers.
Seems counterintuitive. If it's slow, how does the electrical signal manage to get to the entire ventricle quickly enough to ensure a coordinated, powerful contraction?
Ah, that's another absolutely critical role played by that specialized conductive system we mentioned earlier, particularly the Purkinje fibers. The sources tell us these specialized fibers have a much faster conduction velocity, up to 4 meters per second in most parts.
Wow, okay, so the Purkinje system acts like a high-speed distribution network, like fiber optic cables, spreading the signal across the ventricles almost instantly.
That's a perfect analogy. It ensures that once the signal gets past that AV bundle delay and reaches the ventricles, it spreads almost simultaneously to the vast majority of ventricular muscle cells. This allows for that unified, powerful contraction needed to eject blood effectively.
Now, related to that long action potential, there's the refractory period. This is the time when the muscle can't be re-excited, right? It needs to recover.
Yes, exactly. And for cardiac muscle, the refractory period is quite long, and it largely coincides with the duration of the action potential itself. For the ventricle, the sources state it's about 0.25 to 0.30 seconds.
So during almost the entire time it's contracting, you literally cannot stimulate it to contract again, no matter how strong the stimulus.
Correct. And this is absolutely vital for the heart's function. Unlike skeletal muscle, which you can stimulate repeatedly and quickly, causing contractions to add up, and leading to tetanus, basically a sustained cramp, the heart must relax completely between beats to allow time for it to fill with blood. That long refractory period is the built-in safety mechanism that ensures this relaxation phase and prevents potentially lethal arrhythmias that could arise from overly rapid stimulation.
That makes perfect sense. It prevents the heart from cramping up.
Exactly. There is a brief relative refractory period right at the end for about 0.05 seconds where a very strong stimulus might be able to trigger a premature beat, but the main absolute refractory period is the crucial protective window.
And the atrial muscle has a shorter refractory period, doesn't it? About 0.15 seconds, matching its shorter action potential.
Yes, it does.
Okay, so we have the electrical signal, the action potential spreading through the heart. How does that electrical event actually trigger the physical muscle contraction? This is excitation-contraction coupling, right?
Right. And this is another area where cardiac muscle has some really important differences compared to skeletal muscle. The sources describe the action potential spreading along the cell membrane and down into these invaginations called T-tubules, which penetrate into the interior of the muscle fiber. That part is similar to skeletal muscle. And like skeletal muscle, this triggers calcium release from the sarcoplasmic reticulum, or SR, which is the cell's internal calcium storage tank.
And calcium is the universal trigger for muscle contraction, binding to proteins and allowing the actin and myosin to interact.
It is the crucial ion, yes. But here's a key insight highlighted clearly in the sources. In cardiac muscle, calcium also diffuses into the sarcoplasm directly from the T-tubules themselves. This happens when those slow L-type calcium channels we discussed earlier, the ones responsible for the plateau, open up in the T-tubule membrane during the action potential.
Hold on, so calcium comes from outside the cell, flowing in through the T-tubules, in addition to being released from the SR inside the cell.
Yes, and this external calcium influx isn't just important for maintaining the electrical plateau. It plays a direct, critical role in triggering more calcium release from the sarcoplasmic reticulum inside. This process is known as calcium-induced calcium release, or CICR.
Wow. Okay, so the calcium coming in from the outside acts like a trigger or an amplifier to release the much larger stores of calcium held inside the SR. That's a fascinating difference.
It truly is, and it's absolutely essential because, as the sources point out, the sarcoplasmic reticulum in cardiac muscle is actually less extensive, less developed than it is in skeletal muscle. It simply doesn't store enough calcium on its own to produce a full-strength contraction. That calcium entering from the T-tubules is indispensable for activating that full release from the SR and achieving the necessary contractile force.
The sources also mention something about the structure of the cardiac T-tubules being different, larger.
Yes, they are significantly larger in diameter, about 5 times that of skeletal muscle T-tubules. And importantly, they contain these negatively charged molecules called mucopolysaccharides. These molecules bind and store a large quantity of calcium ions right there in the T-tubule space, making them readily available to flow into the cell when the calcium channels open during the action potential. It's like having an extra reservoir of calcium right where it's needed.
Okay. And this whole mechanism, this reliance on calcium coming in from the outside via the T-tubules, explains why the strength of cardiac muscle contraction is so dependent on the concentration of calcium ions in the extracellular fluid, right? much more so than skeletal muscle.
Precisely. The source gives a pretty stark example. If you put a heart in a solution completely lacking calcium, it stops beating very quickly. Skeletal muscle contraction, on the other hand, is much less affected by moderate changes in extracellular calcium because it relies almost entirely on its large internal SR stores. This dependence on external calcium is a fundamental property that professionals working with the heart always keep in mind.
So, for the heart muscle to relax after contracting, all that calcium that flooded the sarcoplasm has to be removed quickly. How does the cell handle that cleanup?
Good question. The sources describe two main mechanisms working together. First, calcium is actively pumped back into the sarcoplasmic reticulum by a specific protein pump called a calcium ATPase, often referred to as SIRC2. Second, calcium is also pumped out of the cell across the outer cell membrane by another transporter called the sodium-calcium exchanger. This exchanger cleverly uses the energy stored in the sodium gradient, which is maintained by the ubiquitous sodium-potassium pump, to move calcium out.
So it's a coordinated effort. Pump it back into storage and pump it out of the cell entirely. And the duration of the actual muscle contraction seems to follow the timing of the action potential pretty closely.
Yes, very closely. Cardiac muscle typically begins to contract just a few milliseconds after the action potential starts, and it continues to contract until just a few milliseconds after the action potential ends. The duration of the contraction is primarily determined by the duration of that action potential plateau, roughly 0.2 seconds in the atria and 0.3 seconds in the ventricles under normal resting conditions.
Okay, this is great. We've covered the muscle tissue itself, the unique electrical signals, and how those signals trigger contraction via calcium. Let's put it all together now and talk about the cardiac cycle, the sequence of mechanical events that constitutes a single heartbeat.
Right, the cardiac cycle. It's the complete sequence of events from the beginning of one heartbeat to the beginning of the next. It's initiated, as the sources clearly state, by an action potential that's spontaneously generated in the sinus node, which is the heart's natural pacemaker, located up in the wall of the right atrium.
And that electrical impulse then spreads rapidly through both atria.
And then, crucially, it travels down through the AV bundle to reach the ventricles. But remember that delay we talked about? The sources emphasize there's a delay of over 0.1 second as the signal passes through the AV node and bundle system.
Right, that built-in pause. That's the mechanism that ensures the atria finish contracting before the ventricles start their powerful squeeze.
Exactly. This delay is timed perfectly to allow the atria sufficient time to contract and push that last bit of blood into the ventricles, acting as those primer pumps before the strong ventricular contraction begins. It maximizes the amount of blood the ventricles have to pump out.
How significant is that atrial contribution to filling that atrial kick?
The source material indicates that atrial contraction typically adds about 20% more blood volume to the ventricles during the final part of diastole, the filling phase.
So you can probably get by without it when you're just sitting around, but it becomes much more important when your body demands more blood, like during exercise.
Precisely. At rest, if the atria aren't working perfectly, like in atrial fibrillation, for instance, the heart can often compensate reasonably well. But during exercise, when the heart rate is high and filling time is shorter, the loss of that extra 20% atrial kick can really limit how much blood the ventricles can pump, potentially leading to symptoms like shortness of breath because the ventricular filling isn't as complete as it needs to be.
Makes sense. Now, the cardiac cycle is broadly divided into two main periods, diastole and systole.
Right. Diastole is the period of relaxation when the heart chambers are filling with blood. Systole is the period of contraction when the chambers are injecting blood.
And the total length of one cardiac cycle is just determined by the heart rate, isn't it?
Yes, it's simply the reciprocal. So if the heart rate is 72 beats per minute, one cycle takes 1 divided by 72 beats a minute, which is about 0.833 seconds per beat. But there's an important point the sources bring up about what happens to the duration of systole and diastole when the heart rate increases.
Like during exercise or stress.
Exactly. When the heart rate goes up, the duration of the entire cardiac cycle obviously decreases. But significantly, the duration of the action potential, and therefore systole, the contraction phase, also decrease, but they decrease less proportionally than the duration of diastole, the relaxation and filling phase.
Ah, so the time available for the heart to relax and fill gets squeezed much more than the contraction time when the heart beats faster.
That's the key point. This becomes a critical physiological limitation. At very high heart rates, diastole becomes so short that there simply isn't enough time for the ventricles to fill completely with blood before the next contraction starts. This ultimately limits how much blood the heart can pump per minute, even though it's beating faster.
The sources provide that really helpful diagram relating the cardiac cycle's mechanical events, the pressures and volumes to the electrocardiogram, the ECG, electrical tracing. Can we quickly walk through how the P-wave QRS complex and T-wave line up with the mechanical events?
Absolutely. This relationship is fundamental for anyone interpreting ECGs. The P wave is caused by the wave of electrical depolarization spreading through the atria. This electrical event precedes the actual mechanical contraction of the atria, which causes that slight rise in the atrial pressure curve you see in the diagram shortly after the P wave starts.
Okay, then comes the QRS complex.
The QRS complex represents the electrical depolarization spreading through the ventricles. Importantly, this electrical event begins just slightly before the onset of ventricular systole, right before the ventricular pressure starts its steep rise. The sources note it typically occurs about 0.16 seconds after the beginning of the P wave, which reflects that crucial electrical delay happening in the AV node and bundle.
Got it. And finally, the T wave.
The T wave is caused by the repolarization of the ventricles as they electrically reset for the next beat. This wave occurs slightly before the end of the actual ventricular contraction, just as the ventricular muscle is beginning to relax. So, you can clearly see how the electrical events P, Q, R, S, T act as the triggers, setting the stage for the subsequent mechanical actions of contraction and relaxation.
Looking at the specific pressure curve for the atrium in that diagram, there are those characteristic little bumps or waves labeled A, C, and V. What do those represent physiologically?
Yeah, those are subtle but informative. The A wave is caused directly by atrial contraction itself. As the atria squeeze that last bit of blood into the ventricles, the pressure inside the atria rises slightly, creating the A wave.
Okay. And the C wave, that happens later.
The C wave occurs right at the beginning of ventricular contraction, during that isovolumic phase. It's caused partly by a tiny amount of blood maybe flowing backward into the atria just as the AV valves close, but is primarily caused by the AV valves bulging backward into the atria due to the rapidly rising pressure inside the ventricles.
That makes sense, the high pressure in the ventricle pushing those valve leaflets back a bit. What about the wave? That's even later.
Right. The V wave occurs near the end of ventricular contraction, while the AV valves are still firmly closed. During this whole time, blood has been continuously flowing from the major veins, like the vena cavae and pulmonary veins, into the respective atria. Since the exit, the AV valve is closed, this incoming blood causes the atrial pressure to slowly build up again. This gradual pressure rise creates the V wave. It then disappears abruptly when the AV valves finally open at the beginning of diastole, allowing all that accumulated atrial blood to rush into the relaxing ventricles.
It's really amazing how much detailed information about the heart's function is encoded in just those pressure curves. Let's turn now specifically to the mechanics of the ventricles themselves, operating as the main pumps.
Okay. The sources break down the ventricular pumping process nicely into the filling period during diastole and the ejection period during systole. Ventricular filling starts as soon as ventricular pressure drops below atrial pressure after systole, causing those AV valves, mitral and tricuspid, to pop open.
And blood that's been waiting in the atria rushes in.
Exactly. That's the period of rapid filling, which makes up about the first third of diastole. All the blood that accumulated in the atria while the ventricles were contracting during the V waves now flows quickly down the pressure gradient into the relaxed ventricles.
What about the middle third of diastole? Is filling still happening?
Yes, but much more slowly. During the middle third, there's just a smaller amount of blood flowing more or less directly from the large veins through the now open atria and continuing into the ventricles.
And the final third diastole is when that atrial contraction happens, the atrial kick, adding that final 20% or so to the ventricular volume.
Exactly. It's the last push to maximize filling. And the sources make an interesting clinical point here. In hearts where the ventricles have become stiff, perhaps due to aging or certain diseases, they don't relax and fill as well during that early rapid filling phase. In these cases, the heart becomes much more dependent on that atrial kick to achieve adequate filling.
Okay, so the ventricle is filled. Let's talk about the outflow phase, systole, when it pumps the blood out.
Right. Systole begins with a really crucial, though brief period called isovolumic contraction. As soon as the ventricle starts to contract, its internal pressure begins to rise very rapidly. This rising pressure immediately pushes the AV valve shut, preventing backflow into the atria.
Isovolumic means the volume inside the ventricle isn't changing yet.
Exactly. For a very short time, maybe 0.02 to 0.03 seconds, all the heart valves are closed. The AV valves are shut below, and the semilunar valves, aortic and pulmonary leading out to the arteries, are still closed above because the ventricular pressure hasn't yet exceeded the arterial pressure. So the ventricular muscle is contracting forcefully, building tension and pressure rapidly, but because it's squeezing against a fixed volume of blood in a closed chamber, the volume doesn't change.
Until the pressure inside the ventricle gets high enough to overcome the pressure in the artery it connects to the aorta or the pulmonary artery.
Precisely. Once the left ventricular pressure rises above the diastolic pressure in the aorta, which is normally around 80 millimole Hg, or the right ventricular pressure exceeds the pressure in the pulmonary artery, around 8 millimole Hg, the respective semilunar valves are forced open. This marks the end of isovolumic contraction and the beginning of the period of ejection.
And blood is forcefully pumped out into the arteries. How much of the blood that was in the ventricle actually gets ejected?
Under normal resting conditions, the sources state about 60% of the blood volume that was in the ventricle at the end of the end-diastolic volume is ejected during systole. This ejected volume is the stroke volume. Interestingly, this ejection isn't uniform throughout systole. It happens in two phases. There's a period of rapid ejection during the first third of systole, where about 70% of the total stroke volume is ejected quite quickly. This is followed by a period of slower ejection during the remaining two-thirds of systole, where the last 30% of the stroke volume leaves the ventricle more gradually.
So most of the blood leaves the ventricle relatively quickly at the start of ejection. What happens right at the very end of systole as the ventricle finishes contracting?
As ventricular contraction winds down, ventricular relaxation begins, and the pressure inside the ventricle starts to drop rapidly. Now, the pressure in the aorta and pulmonary artery, which has been pushed up during systole, is significantly higher than the falling pressure inside the relaxing ventricle. This pressure gradient causes blood in the arteries to momentarily flow backward toward the heart.
And that backward flow is what snaps the semilunar valves shut.
Yes, exactly. The cusps of the aortic and pulmonary valves catch this backflow, causing them to snap shut firmly. This prevents blood from regurgitating back into the ventricles during the subsequent diastole. And the closure of these valves marks the beginning of the final phase of the cardiac cycle, isovolumic relaxation.
Isovolumic again, so volume is constant once more.
That's right. For about 0.03 to 0.06 seconds, all the valves are closed again. Semilunar valves just closed, AV valves still closed. The ventricular muscle is actively relaxing, and its internal pressure is plummeting rapidly back down towards its low diastolic levels. But the volume of blood inside the ventricle remains constant, because there's no way for blood to enter or leave.
Until the ventricular pressure drops below the pressure in the corresponding atrium again, causing the AV valves to open, and the whole filling cycle starts anew. It really is a precisely timed, beautifully orchestrated sequence of mechanical events.
It absolutely is, and understanding the volumes involved in this cycle is key to assessing how well the heart is functioning, something professionals focus on. The volume of blood in each ventricle right at the end of diastole, after it's completely filled, is called the end-diastolic volume, or EDV. Normally, the sources say this is around 110 to 120 milliliters.
And the amount of blood that actually gets pumped out during systole is the stroke volume output.
Yes, that's the amount ejected per beat, typically around 70 milliliters per ventricle at rest. Mathematically, it's the end-diastolic volume minus the volume of blood that remains in the ventricle at the end of systole, which is called the end-systolic volume, or ESV. The ESV is normally around 40 to 50 milliliters. The ventricle doesn't empty completely.
And that leads to the ejection fraction, right? That's a term I hear mentioned a lot in clinical contexts.
It is, yes. The ejection fraction, or EF, is simply the fraction or percentage of the end-diastolic volume that is ejected during systole. So it's stroke volume divided by end-diastolic volume. The sources give a normal value of around 0.6 or 60%. EF is considered a crucial clinical measure of the heart's pumping capability, specifically its systolic function, how well it squeezes. A low EF, say below 50% or 40%, indicates that the heart isn't emptying effectively with each beat, which is a hallmark of certain types of heart failure.
So if someone's EF is, say, 40%, it means their heart is only pumping out 40% of the blood that was in the ventricle at the start of contraction, leaving 60% behind.
Correct. It signifies reduced pumping efficiency. Conversely, the sources note that a healthy, strong heart, perhaps under stimulation like during exercise, can contract much more forcefully and reduce its end-systolic volume significantly, maybe down to only 10 or 20 milliliters. If this is combined with a larger end-diastolic volume due to increased venous return, say 150 or 80 milliliters, the stroke volume can more than double compared to rest. This highlights the heart's tremendous reserve capacity to increase its output when needed, by both filling more and emptying more completely.
We've mentioned the valves opening and closing several times now. They seem absolutely fundamental to making this whole directional pumping system work.
Oh, they are absolutely critical. They are the one-way doors that ensure blood flows in the correct direction through the heart and out into the arteries, preventing damaging backflow or regurgitation.
You have the two atrioventricular, or AV, valves, the tricuspid on the right side and the mitral, or bicuspid, on the left, sitting between the atria and the ventricles.
Right. Their job is specifically to prevent blood from flowing backward from the high-pressure ventricles back into the low-pressure atria during ventricular systole traction.
And then you have the two semilunar valves, the aortic valve leading from the left ventricle to the aorta, and the pulmonary valve leading from the right ventricle to the pulmonary artery.
And their role is to prevent blood from flowing backward from the high-pressure arteries, aorta and pulmonary artery, back into the ventricles during ventricular diastole relaxation. A key point the sources emphasize is that all four of these valves open and close passively. They don't have muscles controlling them directly.
They just respond to the pressure differences across them, like simple one-way gates pushed open or closed by the flow and pressure.
Exactly. When the pressure upstream is higher, they open. When the pressure downstream becomes higher or back pressure increases, they snap shut. The sources note some subtle differences. The AV valves are relatively thin and delicate, designed to close quickly with minimal backflow needed. The semilunar valves, on the other hand, are heavier and stronger as they have to withstand much higher pressures in the arteries. They require a bit more rapid backflow at the end of ejection to ensure they close tightly. They also face much greater mechanical stress over a lifetime due to the high pressures and rapid blood flow velocities, so they need to be incredibly strong yet pliable.
And there's that fascinating detail about the AV valves, the mitral and tricuspid, being connected to these small muscles in the ventricle wall, the papillary muscles, via these fibrous cords called chordae tendineae. What's their exact role?
Ah, yes, this is a really interesting structural feature and a great aha moment from the sources. It might seem intuitive that these papillary muscles somehow help pull the valves closed, but that's actually not their primary function. The papillary muscles contract at the same time as the main ventricular walls during systole. When they contract, they pull on the chordae tendineae, which in turn pull the edges, or veins, of the AV valve leaflets inward towards the ventricle.
So they're not closing the valve, but rather tethering it, pulling the flaps inward to prevent them from bulging too far backward or prolapsing into the atria under the high pressure generated during ventricular contraction.
Precisely. If those valve leaflets were allowed to bulge back excessively into the low-pressure atria, the valve would leak significantly, allowing blood to regurgitate backward. The papillary muscles in chordae act like anchors or guy ropes, maintaining the valve's integrity and ensuring a tight seal during systole. The sources give a dramatic, clinically relevant example. If a chordae tendineae ruptures, or if a papillary muscle is damaged, say after a heart attack, and can't contract properly, the valve can become severely incompetent, leading to massive leakage and potentially acute, severe heart failure. It really underscores how vital these seemingly small structures are for proper valve function and overall heart efficiency.
That small detail about the papillary muscle's actual job preventing prolapse, not causing closure, is a perfect example of getting to that deeper professional level of understanding the mechanics.
It absolutely is. Now, the sources also provide some valuable insight into the pressure changes that occur within the aorta, the body's main artery, throughout the cardiac cycle. We see this reflected in the aortic pressure curve.
Right. As the left ventricle starts contracting and its pressure rises during that isovolumic phase, eventually it exceeds the pressure already present in the aorta from the previous beat, a diastolic pressure, forcing the aortic valve open.
Exactly. Then, as blood is rapidly ejected from the ventricle into the aorta during systole, the elastic walls of the aorta and the major arteries stretch to accommodate this influx of volume. This stretching causes the pressure within the aorta to rise, reaching its peak, which we call the systolic pressure, typically around 120 mmHg in a healthy resting adult.
Then, when ventricular ejection stops and the ventricle starts to relax, the aortic valve closes. But the pressure doesn't just drop to zero, does it?
No, because of the elastic recoil of those stretched arterial walls. As the arterial walls snap back from being stretched, they continue to push blood forward into the smaller arteries and arterioles throughout diastole. This elastic recoil is what maintains a relatively high pressure in the arteries, even while the heart is relaxing and refilling.
I see. The sources point out that little notch, the incisura, in the aortic pressure curve just after the peak pressure. What causes that?
That incisura occurs right when the aortic valve closes. It's caused by a brief moment of backward blood flow towards the ventricle just before the valve cusp seals shut, followed by the sudden cessation of that backflow as the valve closes. It's a characteristic feature related to valve closure dynamics. Then throughout diastole, as blood continues to flow out of the major arteries into the peripheral circulation, the aortic pressure gradually falls, eventually reaching the diastolic pressure, typically around 80 mmHg, just before the next ventricular contraction begins.
And the pressure curve in the pulmonary artery coming from the right ventricle follows a similar pattern, rising during ejection, falling during relaxation, but the absolute pressures are much lower.
Yes, much lower. The sources state the pressures in the pulmonary artery are typically only about 1/6 as great as those in the aorta. For example, maybe 25 mmHg systolic, 8 mmHg diastolic. This reflects the fact that the pulmonary circulation, the blood vessels in the lungs, offers much lower resistance to blood flow compared to the extensive network of the systemic circulation serving the rest of the body. The right ventricle simply doesn't have to generate as much pressure to push blood through the lungs.
Now, all this mechanical action, particularly the forceful closure of the heart valves, creates vibrations that we can actually detect.
Exactly. That's what you're hearing when you listen with a stethoscope, the heart sounds.
Okay. The sources say the opening of valves is usually silent because it's a relatively slow, smooth process. The sounds are primarily caused by the vibrations set up as the valve leaflets snap shut, and the blood and surrounding heart structures vibrate as a result.
Correct. The first heart sound, often labeled S1.
That's the sound produced by the closure of the AV valves, the tricuspid and mitral valves, right at the start of ventricular systole during that isovolumic contraction phase. The sources describe it as a somewhat low-pitched and relatively long sound, often described as the lub, right, the lub, and the second heart sound, S2.
That's the sound generated by the closure of the semilunar valves, the aortic and pulmonary valves, which occurs at the end of ventricular systole as the ventricles begin their isovolumic relaxation. This sound is typically higher pitched, sharper, and shorter in duration than S1, often described as the dub, so you get that characteristic lub-dub rhythm. Listening carefully to the intensity, timing, splitting, and presence of any extra sounds or murmurs between S1 and S2 provides a wealth of diagnostic information to clinicians about valve function and blood flow.
So the heart is constantly doing this complex mechanical work, pumping blood against pressure. The sources also discuss the concept of the work output of the heart. How is that quantified?
Yes, the heart is constantly expending energy to perform work. The work done per heartbeat is called the stroke work output. This work primarily takes two forms. The major component, by far under normal circumstances, is what's called volume pressure work, or sometimes external work.
Yeah.
This is the work done in moving the volume of blood, the stroke volume, from the low pressure of the veins up to the much higher pressure existing in the arteries, aorta or pulmonary artery.
Okay. Lifting the blood against pressure. What's the other smaller component?
The other component is the kinetic energy work. This is the work done to accelerate the blood from a relatively low velocity within the ventricle up to its ejection velocity as it flows out into the arteries. Normally, the sources indicate this kinetic energy component is quite small, maybe only about 1% of the total work for the left ventricle.
But it can become significant in certain situations.
Yes. The sources give the example of aortic stenosis, a condition where the aortic valve opening is narrowed. In this case, the ventricle has to generate a very high velocity to force blood through that narrow opening. Under such conditions, the work required just to accelerate the blood, the kinetic energy component, can become a very significant fraction of the total work output, sometimes even exceeding 50%.
And because the left ventricle pumps against the much higher systemic pressure compared to the right ventricle pumping against lower pulmonary pressure, the left ventricle does significantly more work per beat.
Absolutely. The sources estimate the stroke work output of the left ventricle is about 6 times greater than that of the right ventricle, primarily due to that large pressure difference it works against.
To really visualize and analyze the heart's mechanical work on a beat-by-beat basis, the sources introduce the powerful concept of volume pressure curves and the resulting volume pressure diagram, often called the PV loop.
Yes, this is a really fundamental tool for professionals seeking a deep understanding of ventricular mechanics. It's a graph where you plot the pressure inside the ventricle on the y-axis against the volume of blood inside the ventricle on the x-axis, tracking these values throughout a single cardiac cycle.
And on this graph, you can plot two key reference curves, a diastolic pressure curve and a systolic pressure curve.
Exactly. The diastolic pressure curve shows the relationship between volume and pressure when the ventricle is relaxed and filling during diastole. Typically, the pressure stays relatively low as the volume increases up to about 150 milliliters, reflecting the normal elasticity or compliance of the ventricular wall.
Yeah.
Beyond that volume, however, the pressure starts to rise much more steeply as the ventricle becomes stiffer and is constrained by the surrounding pericardium and other tissues.
Okay. And the systolic pressure curve?
The systolic pressure curve represents the maximum pressure the ventricle is capable of generating during contraction at any given volume. This curve generally rises as the initial volume, and thus the initial stretch of the muscle fibers, increases, reaching a peak pressure at an optimal volume, maybe around 150 or 170 milliliters. Interestingly, the sources note that if the ventricle is stretched too much, very high volumes, the maximum pressure it can generate might actually start to fall slightly, probably because the actin and myosin filaments become stretched beyond their optimal overlap for force generation. The peak systolic pressure a healthy left ventricle can potentially generate is quite high, given its 250-300 mmHg, Hg, while the right ventricle's maximum is much lower, around 60-80 mmHg, though these values can vary depending on the heart's strength and stimulation level.
So if you trace the actual pressure and volume changes that occur during the 4 phases of a single cardiac cycle in this plot, it forms a closed loop, the volume pressure diagram, or PV loop.
This is where the real power of this analysis lies. That loop visually represents the complete mechanical cycle of one heartbeat. And critically, the area enclosed within that loop represents the net external work, or volume pressure work, performed by the ventricle during that specific beat. A larger loop area means the ventricle did more work on that beat. The sources guide us through the 4 phases as they trace out this loop.
Right. Phase 1 is filling. Starting from the end-systolic volume, the bottom left corner of the loop, the volume increases along the low-pressure diastolic curve as the ventricle fills, moving the point to the right until it reaches the end-diastolic volume, bottom right corner.
Then comes phase 2, isovolumic contraction. This is represented by a sharp vertical line moving upward from the bottom right corner. Volume remains constant at the end-diastolic volume, but the pressure inside the ventricle rises dramatically as it contracts against closed valves, moving the point straight up until it reaches the diastolic pressure in the aorta, or pulmonary artery.
Next is phase 3, ejection. This traces the top part of the loop, moving from right to left. The aortic, or pulmonary valve, opens, and as blood is ejected, the volume decreases. The pressure typically continues to rise slightly initially, and then falls as ejection proceeds, following along the systolic pressure curve appropriate for those volumes. This continues until the ventricle reaches its end-systolic volume, top left corner.
And finally, phase 4, isovolumic relaxation. This is represented by a vertical line dropping straight down from the top left corner back to the starting point, bottom left corner. The semilunar valve closes, volume remains constant at the end-systolic volume, and the pressure inside the relaxing ventricle falls rapidly back down to the low diastolic pressure level, ready for the next filling phase. Understanding how changes in heart function alter the shape and area of this loop is absolutely central to advanced cardiac physiology and pathophysiology.
And the sources cleverly link this graphical analysis back to the fundamental concepts of preload and afterload, terms originally borrowed from basic muscle mechanics experiments.
Yes. Applying these concepts to the whole ventricle is essential for understanding cardiac function, especially in clinical settings and disease states. Preload is defined by the sources as the degree of tension on the muscle when it begins to contract. For the ventricle as a whole pump, this is typically considered to be the end-diastolic pressure, or end-diastolic volume, essentially. It's a measure of how much the ventricular muscle is stretched before it starts to contract, determined by how much it filled.
Okay, so preload is about the initial stretch or filling. What about afterload?
Afterload is defined as the load against which the muscle exerts its contractile force. For the ventricle, this corresponds to the pressure in the artery into which it must eject blood, the aortic pressure for the left ventricle, or the pulmonary artery pressure for the right ventricle. The sources often correlate this clinically with the systolic blood pressure the ventricle has to overcome during ejection. Changes in preload, like fluid overload, and afterload, like high blood pressure or aortic stenosis, are incredibly important clinically because they significantly alter the demands placed on the heart and its performance. Understanding these is critical for diagnosing and treating many heart conditions.
Now, powering all this relentless mechanical work requires a constant supply of energy. Where does the heart muscle get its fuel?
The heart muscle is a highly aerobic machine. It relies almost entirely on oxidative metabolism to generate the vast amounts of ATP it needs. The sources state that its preferred fuels are fatty acids. They account for 70–90% of the energy production under normal conditions. The remaining 10–30% comes from other sources like glucose and lactate.
And because it's so aerobic, its oxygen consumption is a very direct measure of how much energy it's using.
Yes, Exactly. Measuring the heart's oxygen consumption is an excellent way to quantify its total energy expenditure. The sources make an important connection here. The amount of oxygen consumed, and thus the energy liberated, is directly related to the total work done, which corresponds to the total area under the systolic pressure curve in that volume pressure diagram we discussed, not just the external work loop, EW, but also an additional area representing the potential energy, PE, which is related to the pressure generated even without volume change. More simply put, oxygen consumption is found to be nearly proportional to the tension developed in the heart muscle multiplied by the duration for which that tension is maintained, sometimes called the tension time index.
This brings in that physics principle, the law of Laplace, doesn't it? Relating pressure, radius, and wall tension.
It does, and it provides another crucial insight into cardiac energetics. The law of Laplace, as applied to a sphere or cylinder like the ventricle, states that the tension, T, in the wall is proportional to the pressure, P, inside, multiplied by the radius r, the chamber, TAPAR. This has major implications. If the ventricle has to generate a high systolic pressure, high afterload, the wall tension will be high, and thus oxygen consumption will be high. But also, if the ventricle becomes dilated, meaning its radius r increases, as often happens in heart failure, the wall tension required to generate the same internal pressure, P, becomes significantly higher.
Wow. So a dilated heart has to expend much more energy, consume more oxygen just to generate the same blood pressure compared to a normally sized heart.
Exactly. This explains why a dilated heart is so energetically inefficient and prone to fatigue and failure. It's constantly working harder, experiencing higher wall stress, even if the external work output, pumping blood, isn't increased. The sources mention that sometimes the heart wall thickens, hypertrophy, in response to chronic high pressure, which can partially offset the increased tension by distributing the force over more muscle mass, but dilation itself is a major energetic disadvantage.
And what about the heart's overall efficiency? How much of the chemical energy it consumes actually gets converted into useful pumping work?
It might be surprising, but the maximum efficiency of the normal heart is relatively low. The source is estimated at only 20–25%. This means that the vast majority, 75–80%, of the chemical energy consumed by the heart muscle is actually converted into heat, not into mechanical work. And in conditions like heart failure, that already low efficiency can plummet even further, perhaps down to just 5%. This highlights the extreme energy crisis that the failing heart faces. It's burning a lot of fuel but getting very little useful work done.
So we've seen the heart's intricate structure, its unique muscle properties, the electrical system, the mechanical cycle, the valves, and the energy demands. How is all of this incredible pumping activity regulated to meet the body's changing needs?
The sources describe two fundamental levels of regulation. First, there's intrinsic cardiac regulation, the heart's inherent ability to adapt its pumping based on the amount of blood returning to it.
Okay, built-in adaptation. And the second level?
The second level is control exerted by the autonomic nervous system, the sympathetic and parasympathetic nerves that connect to the heart.
Right. But the sources also emphasize a really critical overall principle, don't they?
Yeah.
That under most normal circumstances, the heart's output per minute, the cardiac output, isn't really determined by the heart itself, but rather by the amount of blood flowing back into it from the veins, the venous return.
Yes, that's a key concept. The heart, in essence, acts as a highly responsive, permissive pump. It automatically adjusts its output to pump out whatever volume of blood is delivered back to it by the veins. The rate of venous return is primarily dictated by factors in the peripheral circulation related to the body tissue's metabolic needs for blood flow.
And that intrinsic ability of the heart to automatically adjust its output to match the incoming venous return is the famous Frank-Starling mechanism.
Exactly, named after the physiologists Otto Frank and Ernest Starling who described it. The core principle is remarkably simple yet profound. Within physiological limits, the more the heart muscle is stretched during filling, which means a higher end-diastolic volume or pressure, the greater the force of the subsequent contraction will be, and consequently, the greater the quantity of blood, stroke volume, pumped out into the aorta or pulmonary artery.
So, put simply, the more blood comes in, the more blood gets pumped out on the very next beat. The heart automatically adjusts its stroke volume based on how full it gets.
That's the essence of it. The underlying physiological explanation relates back to the muscle sarcomere level. Stretching the cardiac muscle fibers brings the actin and myosin filaments into a more optimal degree of overlap, allowing them to form more cross bridges and generate greater force during contraction. This is a characteristic property of all striated muscle up to a certain point of stretch. The sources also briefly mention a couple of other intrinsic factors. Simple stretch of the right atrial wall itself can slightly increase heart rate by 10–20%. And there's also a nervous reflex called the Bainbridge reflex, where stretcher receptors in the atria can trigger signals via the vagus nerve to further increase heart rate by maybe 40–60%, both also contributing to matching output to input.
The sources often visualize this Frank-Starling relationship using ventricular function curves, right?
Yes, those curves are excellent graphical representations of the heart's intrinsic pumping ability. You typically see two types. One is the stroke work output curve, which plots the work done per beat against the mean atrial pressure, which serves as a proxy for ventricular filling or preload. This curve clearly shows stroke work increasing as filling pressure increases up to a plateau. It also usually shows the difference between the left and right ventricles' work output.
And the other common curve?
The other is the ventricular volume output curve, or cardiac output curve. This plots the volume output per minute, cardiac output, and stroke volume heart rate against the atrial pressure. It shows cardiac output increasing as atrial pressure rises, again demonstrating the Frank-Starling mechanism. As the ventricles fill more due to higher atrial pressure, their end-diastolic volume increases, they contract more forcefully, and thus pump more blood per minute.
Okay, so that's the intrinsic built-in regulation. But beyond that, the autonomic nervous system provides powerful external control over the heart's performance.
Absolutely. The heart receives extensive innervation from both branches of the autonomic nervous system. The sympathetic nerves, which generally act as an accelerator, and the parasympathetic nerves, primarily the vagus nerves, which act as a brake. These nerves can dramatically alter cardiac output, far beyond the range of the Frank-Starling mechanism alone.
So sympathetic stimulation, what happens during exercise, stress, or excitement?
That has major effects. First, it massively increases the heart rate, potentially taking it from a resting rate of maybe 70 beats per minute up to 180–200 beats per minute, or even rarely up to 250, as the sources note. Second, and just as importantly, sympathetic stimulation significantly increases the force or contractility of heart muscle contraction. The sources say it can nearly double the contractile strength.
Wow, so sympathetic activation makes the heart beat both much faster and much harder.
Precisely. This powerful combination of increased rate and increased contractility allows the maximum cardiac output the heart can achieve to increase by 2- to 3-fold beyond what the intrinsic Frank-Starling mechanism alone could accomplish. It provides a huge boost in pumping capacity when the body demands it. The sources even mention that under normal resting conditions, there's usually a low level of ongoing sympathetic tone that keeps cardiac pumping about 30% above what it would be with zero stimulation. So, inhibiting this normal tone can actually decrease pumping moderately.
Okay, and what about the parasympathetic system acting mainly through the vagus nerves?
This has largely the opposite effects. Strong stimulation of the vagus nerves can dramatically slow the heart rate, sometimes even causing the heart to stop beating entirely for a few seconds before it escapes this inhibition and resumes beating at a very slow rate, perhaps 20–40 beats per minute. Parasympathetic stimulation also decreases the strength of heart muscle contraction, but the studies indicate this effect is less pronounced than the effect on rate, perhaps decreasing contractility by only 20–30%.
Why is the effect on contractility weaker than the effect on rate for the parasympathetic system?
The main reason given is anatomical. The vagal nerve fibers distribute primarily to the atria, including the SA and AV nodes, which control heart rate and conduction, but they innervate the ventricular muscle itself much less densely compared to sympathetic fibers. Therefore, their dominant effect is on slowing the heart rate and AV conduction, with a smaller direct impact on ventricular contractile force.
But still, the combined effect of significantly decreased heart rate plus that slight decrease in contraction strength can substantially reduce overall ventricular pumping.
Oh yes. The sources suggest strong vagal stimulation can decrease overall ventricular pumping by 50% or even more, primarily through the drastic slowing of the heart rate.
And the sources show how this autonomic control effectively shifts those cardiac function curves we talked about.
Exactly. Sympathetic stimulation shifts the entire ventricular function curve upward and to the left, meaning the heart achieves a higher cardiac output for any given level of atrial filling pressure. Conversely, parasympathetic, vagal stimulation shifts the curve downward, meaning cardiac output is lower for any given filling pressure. These shifts reflect the combined changes in both heart rate and myocardial contractility induced by the autonomic nerves.
So the autonomic nervous system acts like a crucial master controller, dynamically fine-tuning the heart's performance, speeding it up and strengthening it when needed, or slowing it down during rest, overlaying its control on top of the intrinsic Frank-Starling mechanism.
It's a perfect summary. Now, in addition to nervous control, the sources also discuss the important effects that certain ions in the blood, as well as body temperature, can have on cardiac function.
Right, potassium and calcium ions were mentioned as being particularly critical electrolytes.
They certainly are. The sources note that an excess of potassium ions in the extracellular fluid can have very detrimental effects. It can cause the heart to become dilated, flaccid, weak, and slow down the heart rate. High enough levels of potassium can even block the conduction of the electrical impulse through the AV bundle, potentially leading to heart block. The sources state that potassium levels only 2 to 3 times the normal concentration, reaching 812 mol EQL, can be lethal.
And what's the underlying mechanism for that weakness and slowing caused by high potassium?
High extracellular potassium concentration makes the resting membrane potential of the cardiac muscle fibers less negative. It partially depolarizes them. This might seem counterintuitive, but starting from a less negative resting potential actually reduces the intensity and upstroke velocity of the subsequent action potential that can be generated upon stimulation. A weaker action potential leads to weaker calcium release and thus weaker muscle contraction. It disrupts both the electrical excitability and the mechanical force generation.
Okay, so too much potassium is dangerous because it messes with the electrical potential and weakens the pump. What about calcium ions?
Calcium has different effects. The sources state that an excess of extracellular calcium ions tends to cause the heart to go into a state of spastic contraction. This makes sense, given calcium's direct role as the trigger for the actin-myosin interaction of the contractile process. More trigger leads to excessive contraction.
And conversely, a deficiency to the effect of excess potassium.
Yes, it does. If there isn't enough calcium available, either coming in from the outside or be released from the SR, the contractile process cannot be fully activated, leading to a weak, flaccid heartbeat. However, the sources do make a clinical note that the body usually regulates blood calcium levels very tightly through hormonal control, like parathyroid hormone and vitamin D. So clinically significant problems due to primary calcium imbalances affecting the heart are less common than issues related to potassium disturbances, which can occur more readily, for instance, with kidney disease or certain medications.
That's good to know. And temperature also plays a significant role in heart function.
Yes, quite significantly. Increased body temperature, like during a fever, greatly increases the heart rate. The sources suggest it can sometimes even double the resting rate. The likely reason proposed is that heat increases the permeability of the cardiac muscle membrane to the ions that control heart rate, essentially accelerating the process of self-excitation in the pacemaker cells. Moderate increases in temperature, like those occurring during exercise, can also temporarily enhance the contractile strength of the heart muscle. However, prolonged high temperatures become exhausting for the heart muscle.
And conversely, decreased body temperature or hypothermia.
That has the opposite effect, greatly decreasing the heart rate. As body temperature falls significantly, the heart rate can slow down dramatically, sometimes to only a few beats per minute just before death might occur at very low temperatures, as overall metabolic processes grind to a halt.
So it's clear that optimal heart function is highly dependent on maintaining the body's core temperature within a fairly narrow physiological range.
Indeed it is. Proper thermoregulation is vital for cardiovascular stability.
Okay, one final point the sources touch upon is the effect of the arterial pressure load, essentially the resistance the heart pumps against the afterload on the heart's overall cardiac output.
Yes, this is an interesting and important physiological relationship shown in one of the figures. The sources demonstrate that increasing the arterial pressure against which the ventricle must pump, the afterload, does not cause a significant decrease in cardiac output until that pressure rises to quite high levels, specifically above a mean arterial pressure of about 160 millimilliNH.
Wow. So within the normal physiological range of arterial pressure, say, from a diastolic of maybe 60 up to a systolic of 140 or even 150 millimilliHg, the heart is generally able to maintain its output relatively constant despite changes in the load it's working against.
That's the key takeaway here, yes. The healthy heart is remarkably robust against increases in afterload within the typical range encountered. The sources conclude from this that under normal conditions, i.e. arterial pressure below that very high threshold of 160 millimilliHg mean, the cardiac output is determined almost entirely by the factors that control venous return, mainly the metabolic needs and blood flow regulation in the peripheral tissues, rather than being limited by the arterial pressure itself. The heart, via the Frank-Starling mechanism, simply adjusts to pump out whatever blood comes back to it, largely independent of moderate changes in afterload. That's incredible. We have really covered a tremendous amount of ground here today, diving deep into these sources. We started with the basic two-pump structure and the unique syncytial nature of the cardiac muscle, allowing it to contract as units. Then we plunged into details of the cardiac action potential, especially that long plateau driven by specific calcium and potassium channel dynamics, which is so different from skeletal muscle and so critical for preventing tetanus.
We explored that fascinating excitation-contraction coupling mechanism, highlighting the role of calcium-induced calcium release and the heart's crucial dependence on extracellular calcium entering through the T-tubules.
We meticulously detailed the mechanical steps of the cardiac cycle: isovolumic contraction, ejection, isovolumic relaxation, filling, and the vital role played by the valves, including the often misunderstood function of the papillary muscles in preventing AV valve prolapse. We looked at the volumes and pressure changes throughout, visualized so effectively by that powerful volume-pressure diagram.
And finally, we unpacked the sophisticated ways the heart's pumping is regulated, both intrinsically through the elegant Frank-Starling mechanism that matches output to input, and extrinsically through the powerful accelerator and braking actions of the sympathetic and parasympathetic nervous systems. And we also touched on how performance is influenced by critical factors like potassium, calcium, and temperature.
Yeah, really understanding these core principles, as laid out with such detail in the sources, truly elevates your grasp of cardiac function. It moves you beyond just knowing the heart pumps blood to appreciating the incredibly sophisticated interplay of electricity, mechanics, chemistry, and control systems involved. You start to see the complex machinery and its intricate controls through the same lens that professionals in the field use.
It definitely gives you a completely new perspective on this organ's astonishing capabilities, but also its potential vulnerabilities when things go wrong.
Absolutely. So perhaps a final thought to carry with you from this deep dive, something to mull over. Just consider how incredibly interconnected all these different mechanisms truly are. Think about how a subtle disruption in just one small piece of this complex puzzle, maybe it's a slight imbalance of potassium ions throwing off the heart's rhythm, or a seemingly minor weakness in a papillary muscle leading to progressive valve leakage, or the chronic strain placed on the heart muscle day after day in conditions that persistently increase afterload like untreated high blood pressure. How could such a single issue potentially cascade through the system and ultimately impact the heart's fundamental ability to sustain life? This exploration really shows us not only the remarkable, robust efficiency of the healthy heart, but also highlights the intricate and sometimes quite delicate physiological balance required to keep it functioning optimally.